Yeah, well, we record them and then we put them up for anyone that either wants to go back to something they missed or working on assignments can go back if you showed something that they can do that on their own. And then some of the other faculty that can't make it, they can log in later and watch. So
Absolutely.
Thank you.
Absolutely. We are totally okay with that. Yeah. And I think I have some students in my life joining this this class. Is that okay for everyone?
Oh, that's very helpful. It actually makes for a bigger community. And one thing is the students are logging in. We hope that they stop you and ask questions, because I think that's good for everyone. Because I think sometimes as faculty we get involved and we're used to doing it and don't think to stop and query the students to see what they know or don't know. So hopefully by now, if the students are listening, we expect you to stop them and ask questions. I see a couple of them have logged on, but hopefully they'll do that.
So we recently use Zoom a lot, and not very familiar with it.
Yeah, I think if you want to share the screen, I think you can. Let me see. Yep, you can share the screen.
Okay. Can you see my screen?
All right. So, Noreen, is everyone in there? Is everyone here?
No, I think Sally and Maddie is not here.
Sally and Maddie?
Yep.
I'm here, Sally.
Okay.
Okay. Is Maddie here? Let's give Maddie a couple minutes, if that's okay, Quinn.
Yeah, sure.
Did I pronounce your name correctly?
I want to make sure what kind of a screen you are on your part. You know,
I can see the title slide that says single-cell RNA sequencing data modeling and analysis.
Yeah, what kind of mode is only one page or other pages also?
I see one page right now.
Okay, perfect. Maybe I can.
And what I'm going to do is I'm going to turn my camera off to make my internet work a little bit better.
Yeah, I did the similar thing just now. Yeah.
Okay. All right. So I think Maddie let us know that she's got some she had a personal thing come up that she's going to have to deal with, and she's going to join us when we can. So if you'd like to go ahead and start, that is okay, I believe. And it's recorded, so she can watch it later to get what she missed.
Sure.
I will turn the class over to you, the workshop over to you then.
Thank you, Rusty. No.
You're welcome.
Yeah. So, hi, everyone. And this is Chinma speaking. Yeah. Maybe I can a little bit clarify the pronunciation of my name. Yeah, in the short name, in Chinese, we do not have a K for letter Q. So we always pronounce Q as similar to CH. So Chinma is my name. But if you call my Queen, I can immediately recognize that's me. Yeah, so feel free. Okay.
Start, Chen, can you give us, give the students maybe some background of your training, where you came from before you got to Ohio State?
Yeah, I will. Yeah, definitely. Yeah. So the first slide actually regarding who we are. Yeah.
Oh, perfect.
First of all, is myself. Yeah, I was trained in mathematics. Yeah. Actually, it's applied mathematics. Yeah. During the program, we learned the graph theory, network modeling, the programming, something like that. I went into bioinformatics in 2008. Yeah, when I was a PhD student in University of Georgia. Yeah, my supervisor was Dr. Ying Xu, who was a first-generation bioinformatician in the field. Yeah, and then following graduation, I did my postdoc and the research scientist position in University of Georgia. And then in 2015, I moved to South Dakota State University and established my independent lab. Yeah. Actually, the picture on the left, upper corner is a lab photo. Yeah, when I was in South Dakota. Yeah. And then in 2019, actually the first day of 2019, the lab moved to the Ohio State University. So actually we are kind of new here, about 1.5 years, and so happy to establish collaborations with Kevin, with Jeff, with all of you here, and I'm more than happy to introduce some single-cell data analysis and share some information with all of you. As you can see in this right bottom figure, lab photo, some of the students are also joining us today. I think we have Cankun, Yu Zhou, right? I'm not sure whether other student from my lab has joined this meeting. So Yu Zhou and Cankun, can you introduce yourself a little bit?
Yeah, of course I can. Hi everyone, I'm Yu Zhou Chang. I'm the till now I'm the first year PhD student, but next semester I will go to the second years. And I'm currently enrolling in the Dr. Qing's lab. And my project is more focused on the gene regulatory network inference based on single scRNA-Seq data. And I also control some computer resources in our lab, such as supercomputer computing resource like OIC, Ohio Supercomputers. And I also host the GitHub of our lab. If you have any interest, please contact with Chima or contact to me. I can show some more source regarding our computational tools. Thanks.
Yeah, thank you, Yu Zhou. Cankun, are you here?
Yeah, hello.
Yeah.
Yeah, hello everyone. My name is Cankun Wang. I have been joined Dr. Kumar's lab for about 3 years, and currently I'm work as a biomedical informatics specialist at the Ohio State University Wexner Medical Center. In Dr. Kumar's lab, my research focuses on the development of web servers and databases on single-cell data, and I also have projects on analyzing single-cell data or other kinds of high throughput sequencing data. Yeah, thanks.
Okay. Do we have any other students from our side?
I didn't say.
Okay.
Anyway.
Okay. So if Yu Zhou, if they join us later, so please give him a chance to introduce.
Okay, sure.
And then what do we do? Yeah, basically we are doing bioinformatics and the system computational biology. Basically, we prefer to use computational techniques to bring big data to knowledge. Yeah. More specifically, we are using the high throughput sequencing data. Yeah, and or we just call them sequencing data for short. Yeah. And we want to specifically develop transcriptional regulation information knowledge, yeah, from the data. Yeah, that's the long-term interest. Yeah. And I think including today and tomorrow and the 2 sections in next week, yeah, we are mainly focused on the single-cell multi-omics data modeling and analysis. We will touch a little bit multi-omics data, but we mainly focus on single-cell RNA-seq data, yeah, RNA sequencing data. Yeah. regarding the technology, regarding the data property, we will introduce them in detail in the next 1 or 2 days. Okay, and I showed the website of the lab at the bottom of this slide. Yeah, if you are interested in the research direction, if you are interested in applying a GRA position or internship, and feel free to visit our lab, and also feel free to contact me. Yeah. So I do believe we are not know each other on the table. So I suggest everyone can introduce yourself with your name, your training, and if you have your research interest, please introduce that a little bit. Hi, Rusty, how should we proceed to have a table introduction?
Well, how about I start with myself, and then we will go with our graduate student, the undergraduates.
Sure.
Program. Then
sounds very good.
Then the graduate students, and then the faculty that are in attendance as well.
Sure.
So a little bit about myself. I'm the director for Center for Genomic Advocacy. I'm also one of the PIs of the grant. My background is an ecologist, but I've also worked at the University of Chicago Center for Medical Genetics doing microsatellite instability in colon cancer. We've sequenced wild animal genomes, so we do comparisons there and bioinformatics on that. So there's an interest bioinformatics for some of the species we work with and how some of the genes correlate to behavior. And then I do a lot of work with students, and I think the more I learn about this, I can use tools to answer evolutionary questions I have with my model organism, the white-throated sparrow. So that's enough about me. So I'll turn it over to undergraduates. We'll start with Miranda. Is Miranda on? I can't find the list of who's here.
No,
There we go. Let's see.
I don't see Miranda. Yeah.
Let's start with Dwayne.
Hello, everyone. I'm Dwayne Talley. I'm a fourth-year computer science major. I'm currently in association or collaboration with Dr. Coombs's lab in OSU. And my current project I'm working on is developing an R package to generate a variety of visualizations based off of the cytogenetics of specific cancers like AML.
All right, thanks, Dwayne.
Thank you, Dwayne.
Yeah. Kayla Bennett will be next.
Yeah, I'm Kayla. I'm a CS major. Right now I'm working with Dr. Yan Zhang to do link prediction in biological networks.
Great.
Miriam will be next.
Hello, my name is Marianne, and I am currently working with Dr. Ma with the single-cell analysis by using the Seurat package, and I am a senior undergraduate.
Good job, Marianne.
Yeah.
You did a very good job.
Yeah. Sally.
Hi, I'm Sally. I'm a senior at ISU, and I'm working this summer with Dr. Hugo on data mining, basically with R, doing clustering.
All right.
Thank you.
Excellent. Let's see. I think that's all the undergraduates here. So Noreen.
Sure. So I'm Noreen, fourth-year PhD student. My research project is about an integration of bioinformatics, the bioinformatics prediction tools for enhancer, as well as first I am trying to identify the candidate enhancer regions for the my gene of interest, and then I will verify it from the wet lab. My advisor is Dr. Rusty, and I have serving as a BD for ISU graduate assistant from last 3 years. Thank you.
Yeah, thank you.
And then we'll go in reverse alphabetical order, and we'll start with Dr. Schwab. So Chris Schwab.
Hi, I'm Chris Bob. I am a professor, assistant professor. I do a lot of molecular biology, studying developmental processes using gene expression technology, and then also investigating gene regulatory networks and developing part using fly, and then also human pluripotent stem cells.
Yeah, thank you.
And then Dr. Kyung Cho.
Hi, this is Kyung Cho. I'm an associate professor of biology, and I'm a microbiologist. I'm studying the pathogenesis mechanism of human pathogen, Streptococcus pyogenes, which is usually cause mild diseases such as impetigo and strep throat, but sometimes it can cause very severe invasive diseases such as the toxic shock syndrome and the rheumatic disease and flesh-eating syndrome. So one of the method we are using related to bioinformatics is RNA sequencing. So when you find the important gene which is involved in the virulence of this pathogen, we knock out this gene and then compare the transcriptome profile with the wild type to try to figure out the how this gene globally influence the metabolism or virulence of this pathogen.
Yes, thank you for your introduction, Dr. Cho. I want to have a very quick note, and our lab also have a research interest in microbial data analysis from the basic bacterial genomics like all-prong transcriptome unit regulation, and to the metagenome and the metatranscriptome data analysis. So we have several tools and studies ongoing, and more than happy to talk with you if you are interested in. Okay.
Oh, same here. I'm very interested in your work too.
Sure.
And then it's unusual for him to have to go last because his last name starts with an A, so he usually goes first. But I switched it around today, and Dr. Shad Ahmad.
So hi, I'm Shad Ahmad. I'm an assistant professor of biology here. So I'm a geneticist slash developmental biologist slash systems biologist who studies the development of the heart. And I use both Drosophila as a model system, and working in collaboration with Chris Schwab, I also use human iPSCs directed cardiac lineage.
Okay, thank you so much.
And I believe that concludes everyone that's online right now with us. So I'll turn the floor back over to you.
Thank you, Rusty. Yeah, and if there's a potential collaboration can be developed through this activity, yeah, I'm more than happy to do that. Yeah, and other than that, I will move forward. Yeah, to the content. Yeah, sorry, I used it. Yeah. So this is an outline of the 4-day teaching activity, and basically include what I intend to teach. Yeah, I want to throw out this slide here for a discussion. Yeah, and if something a very eye-catching or badly needed there, I can even change this outline. But so far, let me introduce the plan. This is what I know. Every day we have actually 2-hour teaching with 1-hour break in between. Today, I will first introduce bioinformatics and big data. Yeah, I previously did not plan to teach this part, but I'm not sure whether this part has been well covered previously. So just in case, I put this back. Yeah, and I want to share something with the group. And after break today, we will have a Seurat #1, which means we prepared 2-hour teaching material for Seurat. So this today 3 to 4 p.m. and tomorrow 3 to 4 p.m. we will focus on Seurat, who developed the Seurat and what Seurat can do, and exactly how to use Seurat to analyze your data. Yeah, and I think regarding the Seurat part, Yuzhou Chang, who is my second-year PhD student, will assist and will take in charge of this part. And tomorrow 1 to 2 p.m. I will cover the single-cell RNA-seq data analysis and some pitfalls. Yeah, that's the thing this week. And next week I will keep finishing my introduction of how should we analyze the single-cell RNA-seq data, and then followed up with a pipeline design using Rmarkdown. Yeah. And that part will be assisted by the student whose name is Cankun Wang. Yeah. And last day next week, I will try to have a main theme introduction of how we infer the gene regulation from single-cell RNA-seq data, not general single-cell RNA-seq data. It's kind of a specific topic. Yeah. And along with that, in the 3 to 4 p.m. Next Wednesday, is the IRIS-EDA web server. Yeah, this is an in-house web server developed for RNA-seq data and single-cell RNA-seq data analysis and interpretation. Yeah, very interactive web server. A lot of people use it. Received a lot of attentions. I think that's something we would like to share, and that's something we also recommend you to try. Yeah, if you found that somehow useful to your research, yeah, we are more than happy to see that. So this is the plan so far. Any questions and any thoughts? So please let me know right now. Yeah. Okay, it seems no questions. Okay. If no more questions, let's move on to the goal. Yeah, I always use before after. Yeah, like before the section, I'm confused of how to use bioinformatics and how to analyze single-cell data. So the goal of my section is after the presentation, after my section, you should be still confused. Yeah, and but at a higher level. Yeah, so that's a reality. Yeah, even me, every day I read new papers. Yeah, I learn new things like deep learning and AI, very new things with my student together, and I'm still confused with a lot of things, and we just learn together. Yeah, because this bioinformatics and computational biology, this is a very fast developing field. Yeah, every month there will be new methods coming out. Every month maybe there are several new tools published. We need to keep up to date every month, every week, even every day. Yeah, that's why I think you should be still confused after the 2-week education sessions, but you should confuse at a higher level. You should not confuse at what is single cell and how to write a basic single cell pipeline. You will be definitely able to do that after this 4-day teaching. But you may confuse at a higher level, like for my own project, how should I adjust or how should I optimize my pipeline? Yeah, whether I should use this tool or I should use another tool in my specific data analysis. Yeah, and when I have some data analysis done, how should I interpret those results? Using what kind of visualizations? Yeah, it can give us a much better interpretation. Yeah, those things will come to your mind. Yeah, if you are a graduate student, that's good enough, and you need those thinking. You need those information to write your papers, write your thesis. If you are undergraduate student, think about that. Maybe this is a very good direction you want to go into, yeah, following graduation. You want to go apply for, in your graduate program, yeah. So that's the goal, yeah. And I want to highlight again, this is a group learning. It's not I teach you lesson. It's a group learning, environment. Everybody, you are more than welcome to stop me and ask questions, either during my presentation or after my presentation. Feel free to do that. I want to hear from you. Yeah, I want to see. I want to see somebody challenge me. I want to hear more discussion among the group. Yeah, not like I throw out 25 slides and you can take it or not. That's I think that's not an ideal teaching philosophy. I hope we can learn together. Yeah, I hope we can discuss. Okay. Today, yeah, I'm gonna focus on in the first hour, yeah, and 1 to 2 p.m. Yeah, I'm gonna focus on the topic of bioinformatics and big data. Yeah. So get started with the sentence, yeah, the quote from this gentleman, yeah, Temple Smith. Yeah. I think if you have some literature or training in bioinformatics, you should definitely heard about this person. Yeah. May I ask who know this person and who heard about this person, Temple Smith? Anybody know him? Okay. No, I know some background, and I need more layperson in my future slides. So Temple Smith is kind of recognized as a founding father of computational biology. Yeah, he and another professor, so-called Michael Wortmann. Yeah, they together built the first dynamic programming method for sequence alignment. Yeah, so I do believe everybody should have heard about BLAST. Yeah, BLAST. Yeah, it's a sequence. Yeah, it's a sequence alignment tool. Yeah, full name is basic local alignment searching tool. Yeah. In a long time, people think BLAST equals to bioinformatics because it can enable a lot of analysis in this domain. And if you use BLAST, in BLAST there is a parameter. Yeah, in the parameter show indicating whether or not you will enable algorithm and so-called Smith-Wortmann algorithm. Yeah, actually, Smith-Wortmann algorithm is the very first sequence alignment tool. Yeah. According to Michael Wortmann, he told us he used about one and a half afternoon to develop that algorithm. And Temple Smith is a collaborator to make that algorithm more biological, make more biological sense. So the algorithm was named the Smith-Wortmann algorithm. And I think this is the first fundamental algorithm in bioinformatics. And that's why those two persons were recognized as founding fathers of computational biology. Yeah, I think this sentence regarding bioinformatics, came from his book, published in 2002. Yeah, actually, it's a kind of a soft, definition of bioinformatics. Yeah, this interdisciplinary science is about providing computational support to studies on linking the behavior of cells, organisms, and populations to the information encoded in the genomes. Yeah. So as showcased here in this cartoon, bioinformatics are building bridges among different kind of training, different kind of expertise. Yeah, we are the person standing in the island of computer science, mathematics, and biostatistics. And through bioinformatics, we can develop collaborations with people from animal science, plant pathology, biochemistry, and even physics. So this is that collaborative environment. So that nobody can understand everything in bioinformatics. And that's why I said every day I got confused and I read new things to learn. Yeah. And nobody can have the training and knowledge in all of the elements. But through bioinformatics, we can collaborate. Yeah, we can understand each other. Yeah, I think that's the basic definition of bioinformatics. No matter whether other professors have covered this part, I'm more than happy to share this with the group. Yeah, and it's noteworthy that bioinformatics is firmly positioned in the big data era. Yeah, so big data is kind of, we heard too much of big data. Yeah, too much. Yeah. Like you can easily find such beautiful figures of big data in which information, technology, storage, a lot of things involved, like other statistical numbers. Like big data is everywhere in our human life. Yeah. So what is big data? Yeah. I searched online. Actually, I use this slide in my different teaching classes, and this is so-called 5V. Yeah, some computational people define this like a big data should have these 5 V's. Yeah, I'm not going through every single V, but I will give some examples. So basically, if we describe a big data, it's too big. Let's say too big. It's not a little big. Like if we have a data file with the size of 1 MB, yeah, we cannot say it's a big data. If a data is 1 GB, maybe it's not big data. It's 1 TB, yeah, maybe, yeah, it depends in which domain. But even bigger, yeah, just think about that. Too big, yeah, that's a big data. And second, too unstructured. Which means that the data is not only big, yeah, you need to have unstructured, you don't have a very complicated structures. Like if I have a data size with the size of 10 terabit, 10 TB, but within this data size, all the values are 1, yeah, a lot of ones, duplicated information. You can easily zip this file into a very small size. That's not a big data. Yeah, big, unstructured, and too many different sources. Yeah, that's kind of challenge of big data analysis. Yeah, but I want to highlight the last 2 ways: veracity and value. Yeah. If the data are big, unstructured, but they are junk, we cannot call them big data. The big data should have value to be discovered there. Yeah, should have information and new insights. If we are talking about biological big data, there should have some new biological insights to be discovered in the data. Yeah, such kind of data, we call them big data. Okay. What we can do for big data? Now, this is a very simple slide. I just list 5 bullets. Yeah, we can do data processing. Yeah, we can do data analysis. We can do data management, like data transfer. And the 4th bullet is big data visualization. And the last one was big data modeling. Yeah, I'm not covering every single piece in this big data analysis, but at least as a computational biologist, with a computational training, we can do such kind of things. Yeah, you will see a lot of examples of those big data, analysis or visualization and modeling in my following slides. And we try to have a focus in single-cell data. Yeah, like Seurat. Yeah, we want to introduce this afternoon and tomorrow afternoon. That's a software, yeah, developed by a very reputed group, and they can model big data, they can analyze big data, and most importantly, they can visualize big data in single cell. So a lot of people use that, and now it's quickly grows to a very big community. Yeah, you can see that's a very good example of big data analysis. Okay. Okay, so far, any questions? Yeah. Okay, no more questions. Let's move forward. Like big data, what kind of beautiful patterns can be derived? Yeah, can be identified from big data. Like this is a protein structure example. Yeah, we know RNA have structures. Yeah, we know DNA have structures. Yeah, I think in the previous several decades, people are more focused on protein structure and RNA structure. Yeah, because they have functions. But recently, people are more realize that DNA structure also have a lot of things to do with the functions. Yeah, like folding structure, chromatin accessibility. Yeah, a lot of things here, and those all can be derived from big data. And here is an example of gene expression profiles. If we have I think originally we have macro data, and then we have RNA-seq data, and then we have single-cell RNA-seq data. Anyway, if we have gene expression data, we can derive those beautiful patterns, like a set of genes, their expression have very similar patterns. Yeah, not necessarily linear regression pattern. They can be, similar scaling patterns, like they are going up together, they are going down together, they are very stable together, yeah, but their behavior are very similar to each other, yeah, clustered gene expression profiles. And even more, we can identify networks, a lot of biological networks. Here, we are showing an example regarding protein-protein interaction networks, yeah, but we also have gene regulatory networks, metabolic networks, and a lot of other biological networks. Yeah, if you can define the node and edges, you will get a biological network, and all those networks can be constructed or can be inferred based on big biological data. So here are some examples regarding single-cell sequencing data. Yeah, again, we will introduce the single-cell technology and sequencing technology and the data analysis technology later in this session. But I will showcase an example here. As you can see, we have cell type specific co-expression patterns. Yeah, and if you can see my mouse here moving, and this is a pattern showing a set of genes. Yeah, showcased on the right panel. A set of genes. They are very they have very similar expression patterns, or they are co-expressed in a set of cells, yeah, indicated by the yellow bar, yeah. And another set of genes, they are co-expressed in another cell type, indicating by the purple bar, and then another cell type. So using the single-cell data, we can identify, we can try to infer or elucidate the heterogeneity encoded in cells and cell types. And we're going to identify, enable some analysis which cannot be carried out previously. Yeah. And on the right part is a pseudo-time cell development pattern, or people call that cell trajectory analysis. Using single-cell data, we have the chance to infer how those cells, to infer the cell development and the cell differentiation. Yeah, and especially when we talk about some disease like cancer, like Alzheimer's disease, we will observe what kind of patterns, how cells developed, during the disease development. That's something can be enabled by single cell. There are only 2 examples. Yeah, we will see more examples in how we, for example, how we generate these beautiful figures. Yeah, after this this training, yeah, I guarantee everyone will be able to do that. Yeah. So another interesting topic, yeah, I discussed with my colleagues, with my student several times regarding this topic. Actually, when we talk about big data, it can be roughly classified into two categories. Yeah, we call that big small data and small big data. Yeah. So give you an example of big small data. Yeah. This number was retrieved about 2 years ago. Yeah, like if you say how many transactions finished per day on Amazon, it's about 36.8 million transactions finished on Amazon. Yeah, and just think about this situation. By the end of this year, your manager asks you to analyze all the transaction histories finished in this whole year. Yeah, and you can imagine how many transactions will be there, and that's a huge, the big sample size. Yeah, but in such kind of data, in every single transaction, the information is kind of small or very, very structured. Yeah, like with your the information of your address, the information of your credit card, and the thing you want to purchase, and when you purchase that, and when that delivered. So the information are kind of limited and very structured. Yeah. So it's not hard to analyze this kind of big data. Yeah, definitely not hard. Yeah, we call that big small data. A similar example in bioinformatics data is those behavior outcomes in observational study. Yeah. For example, I have a cohort. Yeah, 5,000 people. Yeah, I have a cohort. I will with some specific purpose. I will take a record of their height, their weight, their heart pressure, whether they smoke or other habits every single week. And after a year, I will accumulate a lot of record behavior outcomes in this observational study. Yeah, 5,000 records per week, and then you have at least 52 weeks in 1 year. That's a lot. Yeah, this is also a typical example of a big small data. Yeah, we have a lot of sample size, but in each sample size we have a very limited number, very limited information. Yeah. Okay. Now we are talking about small big data. Yeah. A typical example is the high throughput sequencing data. Usually, yeah, in the sequencing data, we cannot sequence as many samples as we can. Yeah, I do believe some professors and graduate students in this group, you should have some experience in analyzing or collecting, generating high throughput sequencing data. And because we have a budget, right, and we cannot sequence what we want. Usually we have like 10 samples, yeah, a little bit more, 20 samples. For each of the samples, we will sequence a lot of millions of reads in each of sample. So this is an example of a small big data, which means we have small sample size, but a lot of information in each of the sample. Yeah. So for example, in a typical RNA-seq sequencing, we will have several million short-read sequences eventually. Yeah. And with those reads sequenced, we can get the expression value of every single gene in this genome. Yeah. So we have a lot of information for one sample. Yeah. So this is so-called small big data. And actually, what we will introduce is mainly regarding the high throughput sequencing data. Yeah. And basically, between big small data and small big data, they are not independent. Usually, for example, in the biomedical data, in the behavior outcomes, we will, using a cohort, we're going to secure some very interesting insight. Maybe this specific phenotype are very, very interesting. And then I will pick that specific phenotype and then send it to high throughput sequencing. And based on the analysis of high throughput sequencing, and we can go back to change or otherwise the cohort design. Yeah, so they are connected to each other. Now I throw out this question. Yeah, I throw out this question to this group for discussion. So based on the definition here, do you think single-cell RNA-seq data is big small data or small big data, or not belongs to either of them? Yeah, single-cell data. Yeah, we usually even the very typical SMART-seq2 data, we will have thousands of cells sequenced. Yeah, and usually we have 20,000 genes within the human genome. Just using human single-cell data as an example, yeah, and you will get at least 1,000 of cells in one data. And if you are using more advanced 10x genomics sequencing technology, you may get over 1 million cells. Yeah, that's currently doable for one sample. Yeah, so basically, if we put that into a matrix with each row represents a gene, each column represents a cell, the matrix is gonna be like 20,000 by 1 million. So do you think this data, do you think that this data can be called small big data? Definitely not, because of the sample size. But it's not a big small data. As in every single cell, we have so many information. So I am thinking to create another category here for single cell RNA-seq data. But I'm more than happy to hear from you guys. So any thoughts regarding this question? Yeah, please feel free to share.
So to me, I think it's an intersection of both, right?
Yeah. So it's kind of
you've got
big.
I mean, you're getting multiple reads for each individual cell, and then of course you've got a whole slew. I mean, up to about cells in itself. So
yeah,
to me, it's very much an overlap. But
definitely.
I think you need to define a third category, which sort of makes sense.
Yeah, maybe it's called big data. Yeah.
Yeah.
Big data squared.
Yeah, big data squared. Yeah, I agree with that. So, but at least we are not surprised why the dimension reduction methods are so popular in single cell data, like t-SNE and UMAP, like PCA. Those dimension reduction methods, they get very popular in single cell data because there are a lot definitely high dimensional data. First of all, we need to do dimension reduction. Yeah, we need to select some gene features, or we need to do cell clustering. We need to classify those thousands of cells into 5 cell clusters, or 10 cell types, or 20 cell states. Yeah, and then we can move forward. Yeah, to do the data interpretation, data analysis. Yeah, and we will see those examples in the following slides. Yeah. Yeah, actually, this slide was created by Can Kun. I think he read a paper last night and sent this slide to me. He want to discuss this paper and slide with everybody here. Can Kun, you can jump in.
Okay, thanks, Dr. Ma. So from my experience, often I heard from people that people say like, okay, we have big data, we need to analyze, integrate it, and run some machine learning, deep learning on it. So here, they say big data, they usually mean the terabytes or petabytes of disk space occupied by various omics data like genomics, transcriptomics, proteomics. And the question is, do we really have big data in life science? Like one example is that in recent years, the cost of whole genome sequencing is reduced a lot. So so people may want to know that they can sequence their own genome and predict whether they might have some certain disease in the future. Okay, so and the short answer is that it doesn't work as expected, and one of there are certainly multiple reasons that why genomics has a relatively poor predictive power, and one of the reasons is called the curse of dimensionality, and basically it means that we are enabled to perform meaningful mathematics when the number of features is much greater than the number of samples, since we have a limited number of observations in the genomics data, and sequencing more than 1,000 or 10,000 individuals is not feasible economically or for the majority of the research groups or institutions. So there's the genomic research operates in the ultra high dimensional space, and it's not straightforward to run robust machine learning in genomics, as the amount of data is still a bottleneck. And however, in the single cell studies, and we can see in the figure, this situation is opposite compared to the genomics. Like in recent months, the average cells generated in publication is about 200,000 cells, and so in the single cell study, we can generate a matrix with a gene with a number of features, about 20,000 genes and 200,000 cells. So therefore, I think when we talk about the big data in the life science, so it's about how much statistical power we can extract from this data, rather than how much disk space it occupies, because we can easily generate like petabytes of Gaussian noise, which is absolutely meaningless to analyze. So, and I think the single cell data is one of the ideal big data for machine learning or deep learning to analyze. Yeah. Thanks.
Yeah, great, Sanquin. Thank you for jumping in. I personally like this information, and I totally agree with the conclusion in the title. Single cell data is ideal big data. The size is big, and feature level, which means gene level, we have 20,000 genes, and the cell level, we have a lot of cells. And we can do feature selection, we can do dimension reduction, and the single cell have a very high heterogeneity, which means that it's unstructured, and every single cell may be unique, and they have a different experimental design, a different technology. So we have a batch effect issue, we have a dropout issue, which means we will miss a lot of gene expression data due to the technology capability. We also have the normalization issue. We have a lot of information to be discovered in big data. So based on the definition of 5 V's, this is definitely the ideal big data. And single cell, another point is single cell data is also an ideal big data, can be analyzed by deep learning. And currently, I personally see if you can have a deep learning and a single cell and a complex disease, the paper usually published in very, very reputed journals. And for example, we are doing methodology development. We focus on the Nature Methods journal. I think every single journal every month, you will see single cell or deep learning related studies. And this is a very good opportunity to get data analysis and machine learning, deep learning connected with each other. So more fun on the way. Like I said, I want to share some new information. We are not throwing out the slides we used almost 100 times. When we teach, we will accumulate the most up-to-date information, and I hope can inspire some new collaboration or new insights in the class. Okay, thank you, Chankun, again. Yeah. So it's 1:54. Yeah, I may introduce 1 or 2 slides and then wrap up the first hour. And here said, yes, we are in the big data era. Yeah, we have genomics, epigenomics, transcriptomics, a lot of omics data. Yeah, an interesting reality. Yeah, if you are not aware of this, that's very interesting information. Even now, all the data will be delivered using truckloads, and that's very interesting. Yeah, and I actually tested last month also. It's indeed a similar issue. Usually, when the data is very big, I'm not saying 1 sample or 2 samples, they will send you a link. But when the data is very, very big, the sequencing company are still sending a hard drive to your lab, and then you can download everything there. Yeah, so data are big and too big to transfer using internet. And a lot of information to be discovered. And I threw out this question again. Are we really prepared to fully utilize the data, to analyze the data, to interpret the data, to derive information from data? Are we really ready? Yeah, I'm showing this reality. Yeah, as bioinformaticians and computational biologists, usually it's like we are following the thinking and the research direction established by experimental biologists, like gene. Somebody identified the gene, defined the concept, and then a lot of computational biologists are dedicated in identify genes using data, using computational techniques. Yeah, like protein structure. Yeah, like genome assembly. Those are very typical examples showing what we do right now. Yeah, usually we're following the thinking and hypothesis from experimental biologists. But right now is the reality. Yeah, when the data is small, yeah, when the system is static, the way is good. Yeah, and we really did a good job. Yeah, both sides, experimental biologists, computational biologists, very good. But think about current complex disease system. Think about big data, like. If your boss let you know, tell you, hey, analyze the cancer genomes and tell me why cancer will have cancer, yeah, how to treat cancer, that question is kind of not be easily answered only based on the data itself, yeah. So the traditional way doesn't work, yeah. Actually, compared to a lot of challenges in data analysis and the big data era, and this is my personal view of the major challenge. Yeah, I think to fully utilize the mountains of data to study complex biology, yeah, we need to train or become real data scientists, quantitative data scientists who can understand the true biology. If I can write code, yeah, I can quickly write 2,000 lines of code. That's not real data scientist. We need to learn biology. We need to understand the biological evolution. We need to understand the true biology, and then we know how to ask the right question. And then we are using our computational way to solve that question. I think this training is the main the loss of a real data scientist. Yeah, the missing of this part of training is a real challenge in the big data era, in big data analysis. Yeah, I just want to share my thoughts and share the idea with the group. If some student, either a graduate student or undergraduate student, can be somehow inspired by my talk, and if you decided to train or become a real data scientist, that's the largest outcome of my class. Okay. Rusty, I think it's almost 2. Can we stop here and have a break, then come back at 3? Am I right?
Yeah, that is correct. And I agree with your final point. I think anyone can collect data And oftentimes, and I think there's more of that now, and we're just starting to put these committees together. When we tried to we had a researcher download data from the Cancer Genome Atlas, the secure data from the NIH, and to go through a whole thing. And much like animal care, biosafety, and human subject research, scientists can always collect data, but what really is important is the data you're collecting relevant, and is it answer an actual biological or scientific question? And ultimately, I don't like to use good and bad science, but I think better science is those that have very sound evolutionary questions that can be applied and analyzed, as opposed to those questions I wonder if we collected data, we might find an answer. So it's a pretty good point you make here to end this session.
Thank you, Rusty, for the comment. Yeah, I totally agree with this. Yeah.
So Noreen, you can stop recording now.
Okay.
And we'll join the session again at 3 o'clock. Is everyone agreeable to that?
Yes.
All right.
Okay.
We will
see you guys later.
See you everyone at 3.